So it's a pleasure to introduce our next speaker, Dr. Virginia Moreira Pujol, a postdoctoral research fellow at the Laboratory of Wildlife Ecology and Behaviour at University College Dublin. And today she'll be speaking about the use of Bayesian hierarchical models in INLA and Inlabru for wildlife conservation. So whenever you're ready, Virginia, please take it away.
Sorry, thank you. I'm gonna just share my screen. Just bear with for a sec. Here we go. Right, is everyone seeing the presentation and not the back end of it? Okay, yep. Perfect. Thank you very much. First of all, I would like to say that I might have to not be here for the next speakers after me and for the discussion, and I apologize for that to the speakers and to the people that will be leading the discussion. But unfortunately/fortunately, I have a job interview later that I have to attend, so I will be leaving after this. But at the last slide, there's a QR code with my contact details, so if people have questions that I might not address after my presentation, feel free to contact me. That being said, so my name is Virginia, and I am an ecologist, and I work at the School of Biology and Environmental Sciences here in UCD. And my work entails using Bayesian hierarchical models in INLA and Inlabru as tools for wildlife management and conservation. And I kind of have had to learn how to communicate the very complex ideas that are behind those INLA and Inlabru models to stakeholders, to students trying to learn how to work with them, and to managers that might not be statistically minded. And I kind of gonna walk you through what my work is and how I manage those challenges. And construction has just started next to me, so hopefully you can hear me well. If it gets too noisy at some point, let me know. Right, so my work entails mixing Bayesian statistics, point patterns models, and species distribution models to, as I said, model wildlife distributions and model wildlife distributions in species that are usually of management or conservation concern. That in Ireland, in my case, means mostly deer and badgers, because there are two wildlife species that are involved in human-wildlife conflicts of different types that I'll go into later. But I'm just going to run first through each of these three circles and how they relate to my research, and then we'll get right into it. So Bayesian statistics have been used in ecology, and it seems like when you talk to people, it's something relatively new, but it's not. They're very good for tackling ecological problems because Bayesian models are much more flexible than frequentist models, and they're therefore much better to reflect complex ecological processes. They're also able to incorporate the use of prior knowledge, which is very helpful in ecology because it can help you narrow down some of the responses. And then it's also good for dealing with uncertainty. And it deals with uncertainty with credible intervals instead of p-values, which is a thing that ecologists value quite a lot. Sorry, I'm struggling with the Zoom a little bit here. Here we go. And this is, as I said, not new. This has been discussed, and this paper is from 1996, and it just literally discusses should ecologists become Bayesians, and they have a very strong argument against p-values, as in they provide the null hypothesis that the best statistic would have been more extreme than the actual observed value of the statistic, so it's a probability of data that did not happen, which we all know it's very hard to get students' minds to wrap around the p-values and the hypothesis testing. And maybe Bayesian statistics are a more natural way to speak about these things. I'm gonna quickly sweep through point patterns because I don't think I should be explaining to a bunch of statisticians what point patterns are, but I'm just gonna tell you why are they relevant for modeling species distributions and how I've used them. And here, the picture is from Professor Janine Neely. I learn everything I know about point patterns, which is granted very little from her. So in a traditional species distribution model, you would have a grid over the study area, and then you would study the relationships between presence or abundance of a species in each grid and environmental variables. However, you would not take into account what happens inside that grid. In these two examples, these two grids will look exactly the same for a gridded model when it's very obvious it's not. These models are also scale dependent, which means if you change the scale of the grid, the output will probably change quite a lot. And this is not necessarily exclusive of graded models, but species distribution models tend to ignore spatial autocorrelation. And so the way that I understand and try and explain point patterns to people that are not statistically minded is imagine a gridded approach like the one you see here. These are seabirds foraging off of the Canary Islands, and these are foraging locations. And make the grid smaller and then even smaller and much smaller. And if you keep doing that until each grid cell size kind of tends to zero and it becomes almost one-dimensional, then you have to turn a point pattern, like the one you have on the left of the screen, into an intensity surface, like the one you have on the right of the screen. And the values of the intensity will be related to the density of points, or the number of points that there will be in a grid cell if there was a grid cell in that spot. And if you integrate over this entire surface, this should give you the total number of points in this pattern. This is kind of as far as I go when I try to explain this to people that are not statisticians, because that's kind of as far as I understand it. And that's enough, really. And now we're going to just go through what are spatial distribution models and why they are necessary and how they combine with all the things that I've talked about. And again, there's just a picture of Jane Ellis, because she was and still is one of the people working at the forefront of developing and interpreting species distribution models. So I'm not going to explain what species distribution models are, but I'm going to explain what they are not. So this figure here on the right is the distribution of Sika deer obtained from the National Biodiversity Data Center. And a less statistically minded person would think, right, this is where deer are and are not. But I think we all here probably know that that's not entirely correct. There's a lot of things that affect what this data looks like. Collection or observation bias is a big part of this. It's not, but there is definitely more Sika deer in Wicklow, Donegal, and Kerry. But it's probably also that people go there looking for this particular type of deer because they know they're more abundant there. So these kind of biases that they have to be taken into account, and that's when modeling comes into play. So this would be similar to what I've shown you before. We have a gridded situation, and we have a species that has been detected in these 5 squares. We have looked for it all over the grid, but we might not have seen it, even though if it's there, particularly because animals move. But we also know the environmental preferences of this particular species, and we know that it likes a certain level of precipitation and a certain level of elevation. And so our model will tell us that this particular species is also found in those 2 empty squares, even if we haven't detected it. That's kind of the general idea. Now, how we put those three things together. Well, it's not as simple as that, and that's why I use INLA, integrated nested Laplace approximation. And from now on, we're going to talk about the washing machine paradigm, I think, which is, I don't need to understand how a washing machine works internally to clean my clothes, but I do need to know what each of the buttons do in order to get my clothes properly washed. And I also need to know that this is a very bad idea. So in those kind of terms is the terms in which I understand INLA. And a lot of things that are going to happen from now on this presentation. So INLA is a type of Bayesian modeling. It's not necessarily only spatial, which is what I use it for. You can do a linear model with INLA if you want. It might be a bit of an overkill, but there might be circumstances in which it might be required. But what I use it for is to model spatial distributions of points or patterns of points. And the way to do it is through stochastic partial differential equations. Again, we're going to employ the washing machine paradigm. I don't really need to know what goes underneath the hood, but I do need to know how to parameterize this, because the SPD basically turns the thing on the left, which is mathematically not possible to be integrated, calculated, into the thing in the right. If you define a proper mesh, which is these triangles at the bottom, you will be able to accurately represent the field through this discretized kind of representation of that field. And that's all of the big words that I have for this, and everything else is going to be much more handy wavy. So INLA is a very complicated, complex set of functions, and it has an R package that is just plain INLA, but it's quite hard to work with if you're not very statistically minded. So Inlabru was developed and called Inlabru basically because it was developed in mostly in Scotland. That's as simple as it gets. And Inlabru is a wrapper around the functions of INLA, so that people that are used to work with linear mixed models in R might encounter a syntax that they might be more familiar with and less scared of. And one of the people leading that work was Professor Finn Lindgren, he's single-handedly or almost single-handedly answering most of the questions that people have about INLA and Inlabru, particularly in the Google support group that they have. The problem with Finn, and that's nothing against him, he's a lovely guy, is that this is the way he replies to things. And if you're not a statistically minded person, you read this and you have to dissect it very much to kind of get your head wrapped around it. So an issue with Inlabru that we're currently trying to fix, or well, Janine is, and I'm trying to help her, is to make a documentation that is much more ecology and applied user friendly. But that being said, this is what I use with a lot of help from people that know a lot more about stats than me. And I'm just going to give you 2 quick brush strokes of ecological context of what I use these models for. So deer in Ireland are involved in a human-wildlife conflict because there's a few populations of them that are in expansion, and they're causing a lot of trouble to foresters, to farmers, accidents with cars, problems with native woodlands and biodiversity. And therefore there was a call for a coordinated approach to deer management. And another of the issues is the involvement of badgers in the spread of bovine tuberculosis in Ireland, which clashes with the fact that they are, in fact, a protected species by law. And the amount of money that is spent in BTB eradication and compensation schemes. Right, so we're going to talk first about the badgers, because why keep a logical order? You would think that people would have tried to figure out where badgers are distributed in Ireland or how many are there, but turns out no one really knows. There's not a lot of surveys that have been done. The last one was in the 90s. So we know that badgers are involved in the transmission of TB and they are wildlife reservoir, but we don't know how many badgers are there and how they are distributed. And if they're abundant or their density has an effect on how much of the part they have in the TB transmission. So that's what I tried to figure out. So we have an unknown, which is the distribution of badgers in Ireland. And then I went to the Department of Agriculture, and from then I got the data of where the badgers on the sets were when they went to find them for their culling and their vaccination programs. And I got data that looks kind of like that. That data is obviously very clustered, and that I suspected that that's not necessarily entirely environmentally driven, and that was correct. There is an observation process here at play, and these are the number of days that teams have been looking for sets or badgers in the field in the different parts of the country. And obviously there's a bias there, and I was able to take that into account in my model. Another thing that I needed to consider is habitat preference from badgers. Do they like to hang out in forest? Do they like to hang out in the fields where the cows are, which is very relevant? We know that they like to do their sets in hedges. Do they like muddy, boggy areas, high elevation, low elevation, all that kind of stuff? All of those covariates can be included in an INLA model. Right, there's 1 or 2 slides in this presentation that have a lot of formulas, but the rest of it is going to be just me talking. So this is the model for the badger and the set distribution. There's 1 for each. The estimated number of badgers or sets is the intensity over the entire study area. And this is a log intensity that is something that most ecologists working with quantitative data might be more familiar with than the top bit. So I usually, when I explain this to other ecologists or applied users, tend to skip to this bit. And this has very friendly, familiar faces. We have an intercept. We have covariate effects. In this equation, they are linear. They are not my model. I'll explain later. And then we have a spatial random field that is added, and that's where the SPD comes into play. This is what gives the model a spatial structure and spatial autocorrelation. And this is a Gaussian Markov random field with a mean of 0 and a sparse precision matrix. And I know those words, and I know that that is necessary for the SPD to do its work of turning this field into a discretized version of the field mapped on top of this mesh, triangular mesh. I don't really know exactly why that works. And again, the point is I don't need to, as long as it does, and as long as I know how to fix it when it doesn't. One of the ways that I've said Bayesian statistics are relevant for ecologies is that you can set priors. And for INLA models, the priors are set on the shape of the spatial random field. And the way I set these priors is using this thing that was developed called penalized complexity priors, in which basically what they're trying to do is keep this field as flat, as taut as possible, and let the covariates do the work of observing or reflecting all of the structure in our data. And then this spatial field will represent or observe all of the spatial structure that's not explained by covariates in our data. But the penalized complexity priors are very good at avoiding overfitting. Before these were developed, the usual problem with INLA and Inlabru models would be that your spatial field will overfit to your data, and then your covariates would have no effect whatsoever, even if you did know that the covariates had an effect. And so penalized complexity priors are very good for that. And they are set on the range of the spatial field, and they're set on the variance of the spatial field in a way that you're telling the model that your prior information is that this range is larger than a certain value that you set, and that this variance is smaller than a certain value that you set. So you're setting on the right side of the distribution for the range and on the left side of the distribution for the variance. And that helps you keep this spatial field from overfitting. This is the output, or one of the outputs from the model. This is the distribution of badgers in Ireland. And this is because this is a Bayesian model, for each of the cells of this grid, which are 1 by 1 kilometer cells, we have a distribution. And what is plotted here is the median of that distribution. And to the right is the credible interval of that distribution. So again, this is what I present to stakeholders or to apply to users. I will not show the spatial field, the priors, the penalized complexity, all that stuff, if they don't need to know. But it is useful for them to know that this is the distribution of badgers in Ireland, and that the thing to the right is how credible that result is. So you can see some areas here of larger uncertainty, and then we could discuss with them why that is, and if we need more data collection in that areas. Now, this is a spatial random field. And again, I would show this to stakeholders in the need to know basis, because to me, this makes sense. This spatial random field is, oh, spoilers. This spatial random field is capturing or reflecting the effort, the sampling effort. So clearly, whatever I've done to take into account the observational process has not been enough, because that looks similar to the distribution of the effort. And this is something that I'm still working on, but obviously that's of no help. If it's of no help to stakeholders in what they need to use those results for policymaking and for management, I will not show them that. The thing to the right will be shown to them, and this is the relationship with different covariates. As I said, in my model, these covariates are not set as linear. They are set as one-dimensional SPDs, which is the same model that is behind the spatial random field here, but just in one dimension. And I tend to think about it as something entirely different, but kind of quite similar to a GAM, because of the way it wiggles and the way you control how it does so. And these results kind of show that badgers do not like high elevations, and they like relatively flat slopes. They don't like areas with high human footprint index. They like to spend a lot of time in grasslands, which is concerning because the grasslands variable includes pastures as well. So there's a lot of ecological information we can get from that, this kind of representation of the covariate effect. So this very clearly is something that policymakers and managers need to know. I'm going to run quickly, because I don't want to run over time, over the model that I used for the distribution of deer, just because it's slightly different. It's an integrated species distribution model run with Inlabru as well. So we have an ecological process that is unknown, which is the distribution of deer in Ireland. And we have three different realizations of that process. And we have presence absence data sets from Northern Ireland and from Ireland. They're collected in different ways, different types of surveys, so they can't be put together as one. They have to be two spatially discrete processes, and they are modeled as Bernoulli distributions because they're presence absence data sets, and they have this C-log-log link that. And again, this is the sort of under the hood workings, but having this with the C-log-log link makes it possible to have them at the same scale as a Poisson process here in the right, and with different variants and spatial fields that I'll get into in a sec. And then for the whole island, we had different data sets that informed us of presence of deer. This came from citizen science, from road accidents, from hunters in the north, from different sources with different observational processes, which means we could model all of this difference. So I just put them all together in one data set and hope that the large amount of data would help me with the observational bias and did not add an observational process to this. And this was modeled as a point pattern, so as a Poisson point process with a log likelihood that looks like the one for the badgers. Now, the way this integrated species distribution model works, I model all of the realizations of the model with the same set of variables, because they should be the same things affecting the data distribution, even if the data sets are collected differently, and with the same spatial field for the three of them. Each realization has a different intercept to allow them to be at different levels, which makes sense. And then the spatial field, even though it has the same shape for the three, has a scaling parameter here that allows it, again, to not be exactly the same, just be higher or lower, kind of. As you see, a lot of hand-wavy concepts, but like it's the way that people would understand it if they're not very statistically minded. And again, this is a spatial random field, mean zero space sparse covariate matrix. And this is kind of how I explained the PC priors before ChatGPT had this beautiful way of drawing images where I could do the 4 people pulling on the sheet, which is just like a trampoline. It tries to get the spatial field quite tense. Right. So these are the results for the deer models. The red deer on the left of the screen, the Sika in the middle, and the fallow deer on the right. We see higher populations of red deer in the west, north, and in Kerry. And then we see hotspots of Sika deer in Wicklow and Kerry, which we already knew, but kind of needed to be modeled. And then the fallow deer in the Midlands mostly. Now, the bar here in the right. So what I did for this, if I plot the results in the response scale, that would give me actual values of deer per grid cell estimated. But because the data set was so sparse and so poor, and we could not account for the observational processes, this was a gross underestimation of the actual deer abundances. And as this was meant to be used for management of the overabundance of deer populations, me presenting to them a map that says there's very few deer everywhere would not have been useful for anyone, and especially because it would not have been true. If it's true, that's what it is, and I show them. But it's just a gross underestimation due to the lack of data and the lack of observational process in the model. And so what I did was, instead of plotting this in the response scale, which gives me actual values, I plotted this in the linear scale, which would have this between -5, whatever, and +3, but then rescale it to go from 0 to 1. And the way they explain this to the stakeholders is, even though statistical minds might explode when I say this, there is more likely to find a deer in the yellow areas and the lower likelihood of finding deer in the blue areas. And I know this is not a problem, like it's not exactly how you would explain this statistically, but it's how they understand it best. And it's less confusing for everyone if the goal is to manage overabundant deer populations than if I go with a map that says that there's just very few deer in any area. Another thing that you can look, and in this case, the covariates were linear because it was too complex a model to add even more complexity to it, and in this case, I think it was useful. So I did show them this inset here is the spatial random field, which it's very subtle, but it's much smoother than the thing that you have here in the bigger map, which is the prediction of the model, including the covariates. And so the way I interpret this and I show it to the stakeholders is the covariate effects are very small in most cases. The only thing that seems to be driving the distribution is the distance to forest edge. But everything else, elevation, slope, don't seem to have an effect. Where we know elsewhere in the world, these variables do affect the distribution. And so the explanation in this particular case in Ireland is that Sika and fallow are introduced species, and red deer is in theory native, but most of the red deer present in Ireland now are reintroduced from other places. And so what's driving this species distribution in Ireland is not their environmental preferences, is the place where they were first introduced and then reintroduced. And that's kind of what my spatial field, I hope, is observing a little bit. So the spatial field where the deer were introduced and moved around in the country is represented by the spatial field. And then at the smaller scale in those areas, the distribution is modulated at a short range or a small scale by the environmental covariates, which is what makes this smoother and this less smooth. So that's a way to explain to stakeholders when I am required to show them the spatial field because it's the thing that's explaining my data the most, but they're not going to be able to understand what it is if I go through the SPD and realizations and stuff. So that's kind of like an example of how I show.
Sorry, just 1 minute left there, Virginia.
Say that again?
Just 1 minute left there.
Okay. We've already talked about the confidence interval, so I'm not going to go through this. And there is other ways of evaluating the model with external data that I'm gonna skip. And if people want to know more about this, please feel free to ask me, because I want to dedicate just 30 seconds to this, because I think this is a great forum for this. So in my experience so far, the world of ecology conservation, wildlife management is becoming so much more complex than it used to be, so much more abundance of data complexity, computational capacity, statistical techniques that we're all learning to use, like in the Inlabru, like I did myself. However, biology and ecology disciplines are still surprisingly wary of statistics, and students don't want to take the models, and people don't want to dwell on that and just trying to hide their models because it might scare people. So ecologists have to teach themselves how to do all of these things, and they're always insecure about their skills. So I think, and this is a fantastic place to do it, and this kind of webinar does this very well. We need to build the bridges between the world of stats and the world of ecology, and the previous speakers did that really, really well as well, and to help move the field of ecological statistics forward. And yeah, that's me. Thank you very much. I'm just going to play this. This is a badger, and there's a deer at the back looking at him. And I will take any questions. Thank you very much.
Thank you very much, Virginia. Do we have any questions for our speaker? Okay, I have a wee question. I was wondering, how do you strike that balance, or do you have any advice for especially new researchers of how to strike the balance between like important info to deliver to the stakeholders and what you maybe should hold back so you don't like overwhelm them with information, especially at the beginning, maybe of a research project?
What has worked for me in the past is to get to know the people that I'm addressing and get to know how they react to you showing them equations or complex ideas, and kind of how far along that axis between ecology and stats they are, and then adapting the outputs to that. And then obviously show them everything they need to know. Don't hide anything that is relevant, because that creates a mistrust. Oh, you knew that this was wrong, but you didn't show me. But also don't show them more just to show how much work you've done in the background, for example. So it is a complicated balance, and it hinges on you knowing the other person as much as possible. but I kind of think that's kind of what we need to strive for, to create a relationship of trust between the two kind of sides.
Great, thank you very much. And Simon, do you have a question?
Yeah, just a short one. In your badger example, there was one covariate effect which seemed very nonlinear. I think it was like grass length. How do you communicate that to the stakeholder? Because it appears that it has a great effect in some regions, then small effect, and then a great effect, and then small-ish increasing effect.
Yeah, I don't necessarily trust that much wiggliness myself. So where I would probably go would be look at the overall trend and ignore the small wiggles. But what I would probably do is try and work on that model to see if that is actually a real effect, or if it is
Okay.
just overfitting to some of the small bits of data. Because the data is so clustered spatially that if there's something overfitting, you will find that.
Yeah.
So if I manage to fix that, I'll fix it. But if not, I'll just tell them to look at the overall trend and ignore the wiggliness, and hope that they trust me enough to do that.
Great. Thanks.
Well, thank you very much again, Virginia, and I'll now pass over
you.
to Sylvia for our next speaker.